Separating the major organelles inside fruit fly eggs by density starts with harvesting embryos which are less than 3 hours old. The eggshells are then removed by incubating in 50% bleach. After dechlorination, embryos of defined stages are mounted in agar. As a final step, the embryos are centrifuged and recovered. Examining the recovered embryos using bright light and fluorescence microscopy shows the localization of specific proteins to certain organelles.
A major strategy for purifying and isolating intracellular organelles is to separate them from all other cell components by density. But there's a challenge. When cells are disrupted prior to centrifugation, proteins and organelles that are usually separate may stick to each other under these non-native in vitro conditions. Conversely, some proteins might fall off structures they usually bind to. The main advantage of the technique we are going to describe is that it allows separation by density in vivo. Thus, it avoids breaking open cells and the artifacts that come with it.
This method can help answer key cell biological questions. It makes colocalization experiments very easy and allows the isolation of specific organelle fractions for transplantation experiments and for biochemical analysis.
In this procedure, high percentage agar medium containing apple juice is prepared and poured into plates as detailed in the accompanying written protocol. The plates are used both at the egg-laying step and for the subsequent embedding and centrifugation of the embryos.